Good evening, everyone. I'd like to begin by acknowledging the Bedegal people who are the traditional custodians of the land on which we meet, and pay my respects to their elders past, present and emerging, and also extend that respect to any Aboriginal and Torres Strait Islander people here today. I'm Lyria Bennett-Moses, the academic lead for the Grand Challenge on Living with 21st Century Technology and a Plus Alliance Fellow, and it is in both of those capacities that I welcome you here this evening. UNSW Sydney aims to lead the debate on the biggest challenges facing humanity and has created the Grand Challenges program to foster debate and discussion of these issues. Our program facilitates these critical discussions and in the process raises awareness of the groundbreaking research undertaken at UNSW. It also gives us an opportunity to welcome great minds from around the world. And today I have the pleasure of welcoming a scientist who has made a major contribution to the global revolution in genetics, Dr. Francisco Mojica. Francisco Mojica is a celebrated microbiologist and leader in microbiology at the University of Alicante in Spain. As you'll hear this evening, Francisco discovered and named CRISPR, which pioneered the global gene editing revolution and ranks among the most important scientific discoveries of this century. His cutting-edge research has garnered global attention and earned him a string of prestigious awards, including the Albany Medical Center Prize in Medicine and Biomedical Research and the BBVA Foundation Frontiers of Knowledge Award in Biomedicine. Francisco was also awarded the Plus Alliance Prize for Global Innovation in 2017. He will be joined in conversation by Merlin Crossley, Professor of Molecular Biology and Deputy Vice-Chancellor Academic at UNSW Sydney. Merlin has trained and worked at the University of Melbourne, University of Sydney, Oxford and Harvard, and joined UNSW Sydney as Dean of Science in 2010. He specialises in genetic diseases and recently used CRISPR-mediated gene editing in a landmark study that could lead to new therapies for sickle cell anaemia and other blood disorders. As well as leading a laboratory, he is an enthusiastic teacher and science communicator who contributes frequent articles on science, education, and policy. Merlin will begin this evening with a brief introduction to CRISPR before welcoming Francisco to join him. There will be time at the end for questions, so please make your way to the microphones after the discussion if you'd like to join the conversation. Before we begin, I would like to thank our partners for bringing this event together, the Spanish Researchers in Australia Pacific Network and the Plus Alliance. Thank you, Merlin.
So thank you. I also would like to acknowledge the Bedegal people of the Eora Nation on whose land this university is built. It's terrific to see you all today. I've had a wonderful day with Francis. He's worked very hard doing interviews, and he gave a seminar in the School of Biotechnology and Biomolecular Sciences, and it's the first time I've ever seen the students come up and ask for autographs at the end of the seminar. It was quite something. So I've just got 5 slides. The first one, I do want to show a picture of the Alicante, which I think has the second most beautiful harbour in the world. And I think it is important actually that the work we're going to describe began in Alicante. And I think the diversity of scientific thought, the best ideas come from all around the world, and science knows no boundaries. So I wanted to start with that. Then I just wanted to give my perspective on what CRISPR is all about. So to me, CRISPR is like Google. Now, anyone who's my age would remember that's a picture of what our libraries used to be like. And you could find things in the library, but it was enormously difficult, painstaking, and it took a long time, and you needed a librarian to put everything in order. Our genome is not in order. And our genome is like our library catalogue. It is massive. 3,000 million letters in the genome. But it gets worse because humans are built not like Wallace and Gromit there. They're not like that. They're more like that little fox made out of Lego. We're built out of 30 trillion cells. But the cells are such that every one of them is like a smartphone with its own computer code. And all our DNA is in every cell. Now, this is important because it tells you how much DNA we have, which is massive. It's sort of to the moon and back if you stretch it out. It's massive. But also, it tells you, because we're built like that, when you're modifying things with CRISPR that I'll come to, you've got to get it into every single cell. And that puts a limit on what we can do. So it's like Google, because CRISPR can now find any gene. And so these days, you type into your Google search. What we do in the lab is we make an RNA guide in green. RNA is just like DNA. It's just a slightly different chemical, just missing an oxygen, or having an extra oxygen. And the guide and these proteins in orange will search through our entire genome until they match up by, you've all heard of Watson and Crick base pairing. And that is what CRISPR is. And then once it matches up, it cuts the gene. And that's the secret, and that's all there is to it in a sense so far. Although, from what I heard, Francis is doing much more work, and we're discovering more and more about this every day. So you can use it to cut a gene and put in a new gene. And that's what everyone's talking about, modifying genes. But also you can just use it for detecting DNA. And so here, when it finds DNA matches the RNA guide, it cuts. And you can make detectable markers which are released. And there's something called Sherlock, built at MIT, Broad Lab, Harvard, and Detector in Berkeley. And you can see you can detect DNA in cells, blood, someone's throat, and in the bottom there's urine and some mud there as well. So you can detect DNA in anything. And that really is the secret of CRISPR, that it can find any gene in our genome, it can cut it. And it is Francisco who discovered this. And he gave it this funny name, clustered, regularly, interspaced, short palindromic repeats. And you look at that and you think, well, that doesn't tell me much, but it told him a lot. There are lots of repeats in DNA. Most of them are random junk. But Francis had the foresight to realize this is not random junk. This is a pattern and this is a system. And he worked out the repeats matched bits of viral DNA, and he worked out that CRISPR had evolved in bacteria to cut invading viral DNA. And once he worked that out, the rest of the world worked out, if that's how bacteria cut DNA, we can use it to cut DNA too. And that is the beginning of the second revolution in recombinant DNA technology. So without further ado, I invite Francis to the stage. We'll have a conversation for half an hour or so, then there'll be questions from the audience. Welcome, Francis. So Francis, you love biology and you love bacteria. Can you tell us a little bit about what life's like as a bacteria? As a human in Australia, we're worried about being hit by a shark or being hit by a bus. What do bacteria worry about?
Bacteria worry about these small, tiny viruses that attack them and try to destroy them almost every second. On Earth, you get a killing of bacteria by viruses of about 50% of all bacteria every 2 days, which means that, in fact, the viruses control the bacteria in the world in general. And in some places, you have about 10-fold more viruses than bacteria that are trying to kill them. So they have to defend from that attack.
And what are viruses?
What are viruses? Viruses are not living beings. Some people say they are living beings, but they are not. They are, from my point of view, they are genetic elements made of part of this information, a little bit of information of DNA or RNA that manage to get out from the bacteria and move from one bacteria to another, protected by proteins that make an envelope. So they are genetic elements, mobile genetic elements that are stable outside the cell. They need the bacteria to reproduce. The only thing viruses pursue is just to get more copies of themselves and infect other bacteria. It's very strange that some organism just thinks about reproduction. Just reproduction, that is what they do.
And so they're like computer viruses in a way? Are there analogies between viruses and computer viruses?
Not at all. But anyway, they infect things and they destroy things, so that's the relationship between the real viruses and the computer viruses.
So tell me about when did you first notice CRISPR and when did you first notice it was an antivirus system?
Okay, nice story. I was working on my PhD thesis work. I was a bit younger than now. I had a lot of hair on my head. And I really enjoy doing some research just because you like knowing how biological systems work. We had a question. Okay, it was not my question. Someone told me, this is your question that you have to solve and you have to get an answer to that. That was my PhD thesis advisor. I don't know how it works in Australia, but in Spain, when you try to do some PhD thesis work, is the advisor who tells you what you have to do, at least at the beginning. After that, maybe you follow that direction, but the first direction is that. So I was told you have to try to know which is the reason and the regulatory mechanisms involved in adaptation to high salinity by these microorganisms. They are not bacteria, but very similar to bacteria. And I found these regularly interspace repeats, this CRISPR. And also we noticed that there was some report published a few years before where they found also regularly interspace repeats in a completely different microorganism. So I was interested in knowing why these sequences were present in such a distantly related organisms that had been evolving separate for 3,000 million years, something like that. And after my PhD thesis work, when I finished it, I planned to just focus my research on these repeats, trying to understand what they were doing. That took 10 years, 10 years of work. And after 10 years, we just found a sequence in between these repeats that derive from a virus. And we knew that this particular isolate carrying a sequence matching another sequence for the virus were resistant to infection. And after that, we analyzed many other CRISPR from many other organisms. And we came across an immune system, an adaptive immune system. We got the proof of something that was really unexpected. You think about that, it's just amazing.
Yeah.
So bacteria are able to recognize something that is trying to kill them and keep a memory of that, a genetic memory of that.
Which is a very important defense. So you worked on it for many years and you kept digging and digging and digging until you hit water. You never gave up and decided, I need to work on what everyone else in America is working on?
Almost every day. Probably I was not able to find anything in microbiology, that is my specialty, that could be more interesting than these sequences. That's the reason. So I was looking for an alternative, another kind of project or whatever, but couldn't find anything that could be more interesting. I think it's not good to follow the trends of people. Something is fashion. CRISPR is fashion now. It's trendy, and people like to work on CRISPR. I think it's good to work in looking for other lands, other ways. And this was new. No one else was interested in CRISPR, so this is just for me. So I can take all the time working on that. There's no competition there. And I'm a professor, so I'm getting paid just for teaching. And research in Spain is like a hobby. So I spend my free time doing some research. It's a very nice opportunity to do research without pressure. Probably there's some research people here and know what I'm talking about. Pressure on research. That's not a good thing.
And did a group of students who had a similar philosophy join you in your research?
Yeah, I don't know why. I still they trusted me. They were just desperate, or I don't know. But not many people. One. It's from one guy, and after that I convinced a second one.
And when did you first realize that people were going to adapt these ideas to translate them into tools for recombinant DNA work?
I remember that day. That was in 2012. I was in my office doing some funny things. I don't remember exactly what. And one of my collaborators just came into the office and said, "Francis, look at this. These people says that they could use this tool to edit the DNA of any organism." And I said, "Okay, I have so many things to do now. Just we'll talk about that later." And I have to confess, I couldn't believe that at that moment. In fact, I'm a microbiologist that works with bacteria, and we know that when you cleave DNA from bacteria, usually they die. I was not completely aware that when you cleave DNA in a eukaryotic cell, that provides you the opportunity to edit the DNA. That was the beginning of this toolbox of CRISPR, as you mentioned. Just genome editing was one possibility, and now we know that this is not just the only one. There are many other applications of CRISPR.
Yeah, that's right. We should say that people had worked out that when you cut a human gene, the cell will grab any DNA can get and stitch over the gap with it. And people were able to do this with things called zinc fingers and talons, except it was an agony to do because it took months and months and months, and most of the time didn't work.
Yeah.
But CRISPR works remarkably well. So now tell me about, do you have any advice you'd give to young people today who are interested in science?
Oh. Don't do what I did. Yeah, you have to stop at a certain point. You cannot afford 10 years of research looking for something when every single hypothesis becomes wrong and all the experiments are wrong. You can afford that when you have a position like the one I had, but not many people have that possibility. If you are doing your PhD thesis work, you have 4 years here in Australia, 3 years in Spain. You have to get results. This is probably too risky. I would advise that you have to combine both things, very risky things, and things that will provide you and produce some results for sure. So that way you will enjoy science with the risky things, and also you get some papers. You have to publish papers. If you want to get a fellowship, want to get a grant, and you want to do to keep doing research. So I don't know. I'm not a person to give any advice, because myself I think I made many mistakes, and I was lucky with CRISPR. Just that, I was lucky. So I wouldn't recommend people in general to spend much time trying something that doesn't work. So, but any contribution to knowledge will give a benefit to the society for sure, and that's a good advice.
So I think that's the perfect advice. I say to all my PhD students, you should have one safe project you can demonstrate you're productive, like salinity, and one which you're exploring the unknown. And it's easy to have those two projects as long as you work twice as hard as I had to work. So I think that it is very good. It is very good advice. I want to ask you about what you think CRISPR might be used for in the future. So recombinant DNA has been with us since the 1980s, really, was the beginning. Originally there was a moratorium, but people quickly realized that it wasn't, there has been very little harm to the world or individuals by recombinant DNA so far. What do you think about the future of recombinant DNA work now that we have a tool which is much more powerful?
With CRISPR, the knowledge on the role the different parts of the genomes are playing is being much easier to understand. That means that in I think 1 decade, 2 decades, probably the personalized medicine could be a reality. That means that it could be possible that every single disease will be treated in every single patient in a different way to avoid any side effects, because we will know exactly which is the effect or the role played by every single part of the genome. And also with CRISPR, it's expected, that's not real yet, we don't have proofs of that. It's expected with CRISPR also, thanks to their knowledge derived from the use of the CRISPR technology. With CRISPR also you can apply them as a therapeutic agent to cure diseases. That's a promise. There are about 20 clinical trials now planned for the near future, or ongoing clinical trials in humans to cure diseases. But we don't have any result yet. So maybe we will be used as therapeutic agents, and maybe we can get rid of some infections by viruses or some genetic diseases. But we'll see. From my point of view, all these clinical trial experiments are just experiments so far. Just today, I knew that the 2 papers have been published saying that the consequence of modifying DNA with CRISPR could be even to produce a tumor.
Yes. Yeah.
Okay? And We just have known that now, 2 years later, after the first clinical trials. And also a few months ago, we knew that many people have antibodies against Cas9 that could produce some side effects when a patient is injected with CRISPR-Cas9. And probably there are many other things that we don't know yet. So we had to be very prudent on that. And many people with diseases, genetic diseases, send me emails asking me about the possibility of her son or her daughter with a genetic disease to be cured. And I said, maybe. But the situation right now is that maybe, we don't know.
So today in your talk, you showed a paper about an evolutionary biology paper where people have compared lizards to snakes. There's a particular part of the genome that has been lost, and that's why snakes don't have legs. Mm-hmm. And they also have now modified the corresponding genome in the mouse and produced a mouse that has no legs, but it has none of the other benefits a snake has. So that in a way is a striking but quite a shocking experiment in many ways.
Yeah, it is.
Do you think that there will be more modifications to agricultural animals and plants?
Yeah.
Will be faster than human disease?
Absolutely. As a proof of concept, now a few weeks ago in the States have said that they are not going to regulate these plants for human consumption when they are modified with CRISPR.
Oh, that's right.
Yeah. And Europe is saying the same thing. It's not stated so far, but they just said that probably they will go exactly in the same direction. Because when you modify the genome of a plant with CRISPR, you are just doing exactly the same thing that happens in nature. But instead of waiting for hundreds of years or just selecting among millions of plants to identify the plant with the right mutation, you can get that mutation in just a few weeks. So that's the difference. It is not a transgenesis. You are not introducing foreign DNA. You just use the scissors to cut, edit, and that's it. And the tool disappears. So it doesn't remain in the cell. So it's exactly the same that happens in nature. So now there are many plants that have been modified with CRISPR to make them resistant to abiotic stress, to biotic infections, resistant to any, for instance, any pesticide or things like that, to get them grow faster and to give more nutritious fruits. And so I think the field of agriculture in general will be the first to benefit from CRISPR rather than medicine. Everyone is talking about medicine. When you are talking about health, that's the most important thing. You know that?
To everyone.
Yeah. So, but that's not really, from my point of view, the most sure output of CRISPR. Agriculture.
Yeah, I agree. I think the first beneficiary is the scientists. We could do a lot more.
Yeah.
The second beneficiary will be agriculture. And human health, there will be the greatest investment in human health, but it will take time.
Yeah, yeah.
And I agree with you also about this. Recombinant DNA, there was always concern because often you were adding a gene from a virus into a human, and there was anxiety that that would be a problem. Often, mostly it wasn't, but this you're right. It is simply, you aren't adding anything, you're just making a change to the spelling, or sometimes you might be deleting a gene that's not good, and people don't have the same ethical concerns with this. There was another revolution that never took hold. When Dolly the sheep was cloned, there was a great enthusiasm that people would be cloned, and that didn't happen. Then there was the idea that pet dogs would be cloned, and that happened like there's one or two, but it was too expensive and it was too difficult to do. So I think also the uptake of some of this won't be as great as some people think it is. It would be. So it'll be slower, and there will be time to discuss the ethical issues as we go forward with this. So what is your thought then? Do you think overall this technology will be used for good, or do you think there's aspects of it that we need to be very cautious and aware of?
Yeah. Okay, most researchers are responsible for what they are doing, so will be applied for good for sure. With this technique, you can afford any change in any cell of any organism quite easily, and this is very cheap. CRISPR has democratized the genome editing, so almost any lab, even non-molecular biology labs, can and are using indeed CRISPR to edit genome. That means that everyone in every part of the world can do that. And some people has a very strange mind and way to think. So you can never guarantee that someone is not going to do something wrong. So the possibility is there. But as you mentioned, there are many examples of tools, if you want, or ways to do something wrong. When you realize that's possible, all concerns come to your mind, and people just used to make summits and meetings trying to discuss on these sort of things, and after a few years nothing happens. I hope this will be the case. But this, of course, that's the price you have to pay when you have such a powerful tool. All the good things of the tool, it is easy to use, it's cheap, it's very efficient. All these things make also to think about the dark side of the tool.
And do you think, in your seminar today, I learnt that there are so many other antiviral systems that bacteria have, and there are so many different, we'll use CRISPR-Cas9, but there are many, many different Cas proteins, and we know Cas12 and Cas13 does RNA, and there's, how much more do you think there is to be discovered simply by adapting the current tools?
About CRISPR systems?
Yeah.
I will give you one example. We only know about from 0.1% to 1% of prokaryotes on Earth, because we have to cultivate them in the lab, and only a tiny proportion of prokaryotes are able to grow in artificial conditions in the lab. And all we know, all the tools you mentioned, and many more derived from CRISPR, are thanks to the knowledge of these very few cultivated microorganisms. In a very recent paper, they looked at nature, taking samples of nature, checking DNA, sequencing, all the information there, and they found 3 different CRISPR-Cas systems that are completely different to those that we know. 3 different. One of them relies on only 2 genes, that means 2 proteins. And others are relying on 3 or 4 proteins. And when you compare these proteins to those of the known CRISPR-Cas systems, probably they are working in a different way. So we could find 100-fold more CRISPR systems than we know right now. And that's related to CRISPR. But you already mentioned also that 1 month ago, someone found 10 different antiviral systems. So this is very, very, very heavy stuff. You have bacteria fighting with viruses, viruses trying to protect from these antiviral systems, this arms race that has been running for thousands of millions of years means that there are many, many hidden tools in nature, and probably we'll get some benefit from that. And I hope that in a few years we will not be talking about CRISPR, because probably other tools and other systems will be better, and CRISPR could be superseded by these other systems. That would be a very nice new.
Yeah. I mean, we have come a long way. Realizing that DNA was digital was the first step. That the double helix is something, but the fact it was linear digital, the fact that we could modify it at the beginning of the recombinant DNA world, but now that we can edit any base is huge. And the fact that bacteria have been editing the DNA of their foes for so long gives, I agree, there's great prospects. We could do more. Now, I'm just going to ask you one more question, and then we can go to the audience for questions. There's a couple of mics, but the question I want to ask you, you gave some advice reluctantly to students about how to manage their research. Wondered if you had any advice for university managers about, about how to manage the research productivity of the university. Should we let everyone work for 10 years on every problem, or what should we do? Because it's a constant challenge.
I don't have an answer to that.
You're not the only one.
I can complain about many things, of course, but researchers are not, how you say, multifunctional people. And we have to do, at least in my case, teaching, research, taking care of papers, filling papers all day, trying to justify projects. Okay, I'm not going to get you bored about this, but there are so many different things. I think it's important. It's important, for instance, in my case, to do teaching and to do research, because you know much more teaching than just doing research. And that's important. To have a general view of microbiology, in my case, helps a lot.
Yeah.
But that's it. I mean, I don't have time. I shouldn't spend my time doing something different from that. And I don't know in Australia what's going on, but in Spain, every single year you have more things to do just because of this sort of papers you have to fill, reports and everything. We are not specialists in filling papers. I hate filling papers.
Do you think we need an anti-paper defense system to protect us from the bureaucracy that's ever growing?
Yeah.
I think we may evolve such a system, but I'm not sure. So look, it's 1:15 and we've got about 15 or 20 minutes for questions. There are some microphones, but it is so dark out there that I can't see. There's one here, and there'll be another one in the gloom just over there. I can see it. And if people would like to come up and ask a question, but I ask you to come to your stand up and walk to the microphone, introduce yourself, and please ask a question. Well done, our Dean, leading by example.
Hello. Oh. Hello, can you hear me?
Yeah.
Yes. Emma Johnson, Dean of Science here at the university. Welcome and thank you for joining us this evening. It's been delightfully frank conversation. I have a question born from my ignorance of the new technique. How accurate is this gene editing? Everyone's talking about it as if you can get into the single nucleotide, you can get right in there. But how accurate is it, and what's the error rate around these cuts? What are the consequences of those errors?
Okay. You probably know much.
You-
Okay, no problem. You can design a guide, this guide RNA, to direct the Cas protein to cleave in a sequence that perfectly match the guide. Okay, that means that you can, in theory, drive these scissors to a single location in your 3,000 million letters of your DNA. But the Cas9 or the Cas protein, these scissors are also able to cleave sequences that are not 100% identical or complementary to the guide. These are called off-targets. The native Cas proteins, the native scissors, tolerate up to 5 or 6 mismatches. That's a lot. That means that there are other places in your genome that will be cleaved and edited in a random way, in an unintended way. But now researchers have developed variants of these scissors that are high fidelity, and they don't tolerate any change in the sequence. They only go to the right place that is 100% identical to the guide. There are also some worries about the fact that sometimes you get a change, an unintended change in the intended location, without not off targets, but on targets. And that's also a problem. That's a problem, for instance, when you transfer these tools to a reproductive cell, to a zygote, then you get changes in some cells that are different from changes in another cell. And you can get a mosaicism in the organism when it grows. That's another worry. There are many concerns and many drawbacks of this technique. They are being solved, but probably the limit will be the knowledge. Once again, knowledge is very important in applications, basic knowledge, the knowledge of the repair systems. So mistakes and wrong editions are the consequence of the fact that eukaryotic cells have several repair systems. And some of them, and in fact are the ones that are working most of the time, make random changes. You can direct exactly the change you want to make, introducing in the cell, apart from the scissors and the guide, a fragment of DNA that will repair that region after the cleavage. But you still have the other system there that is making random changes, desperate to repair that break. And in those cases, you can get unintended changes. So about off-targets, people was very worried at the very beginning when the first genomes were edited with CRISPR. After a while, they said that's not really a problem, because also the frequency of these off-targets, of these mutations elsewhere, is about the same that happens in your cells when they divide. So it's not a big deal. But for a therapeutic application, of course, you have to get a situation where the unintended changes, the side effects, really justify the use of these tools or any therapeutic agent. That happens with any drug to treat any disease. But in particular with CRISPR, we have to be very, very careful with that because of putative side effects. And also when you change a fragment of DNA, the sequence of a fragment of DNA, just because of our ignorance on the effects of changing a piece of DNA with other places on the genome, you cannot predict always what is going to happen.
I mean, I think it's pretty good what happens if you when we do it in blood cells, if we have 10 blood cells, 3 of them might be right, the other 7 aren't right, we throw those away and work on the ones which are. But the systems are getting better and we're learning what works and what doesn't. And the off-targets, people, just as Francis said, people were very concerned, but they're making better and better CRISPRs that I think it'll be all right. It would only be for human germline modification that you'd be really concerned about this. I think even for somatic mutations, you'd probably, if it was a serious disease, you'd had fewer side effects for this than most medicines. So I think it's all right. So, but people will keep asking that question. Rob.
Yes, Rob Young from BABS. So I have 2 quick questions. One is that I happened to come across 2 papers this morning or maybe yesterday from Nature Medicine, which showed that the efficiency of CRISPR in stem cells, the work doesn't work very well because of P53, right? P53 is the guardian of the genome.
Yeah.
So apparently the stocks of 3 CRISPR-linked companies have crashed today. So I'm just, my question is just, what's your view on that, right? In terms of application of this technique, eventually, hopefully, we will do it in stem cells, right? To modify the genome to help, to make to.
Yeah. This is, in fact, the paper I was mentioning before, the P53 issue. Yeah, that's one of the things that justify that we don't have to rush. We don't have to go too fast, just because we are getting some results, unexpected results as these that are telling us that sometimes you are getting side effects as this that can be very, very, very bad for the cell, very bad for the research, very bad for the therapeutic application of CRISPR. As far as I know, I'm not an eukaryotic researcher, but I was reading the commentaries on this P53, and they really say that you can get even tumors after the application of CRISPR, just because this you will select some cells that have a reduced production of P53, that this is the guardian of the cell. So in that case, working all the time is the same thing, working with cells of the germ line or working with stem cells, you get that problem. You can get from that many cells with no accurate control of the cell cycle and things like that. This is one of the problems that I guess will appear in the next months or years. We don't know everything, how the consequences of genome editing. This is a very important one. And the other one was the previous one, that the one I mentioned that is, I don't know whether I mentioned that here or in Japan or China, anyway, is that we have antibodies against Cas9.
Yeah.
So these are 2 examples of our knowledge on the consequences of genome editing.
Yeah, we don't know, but it might be a good time to buy stocks if they've crashed, because there may be a,
now
there may
low.
be a way around.
Yeah. Sorry, I have another question that you do not have to answer. I want answer from each of you, actually. This CRISPR will win someone a Nobel Prize. I want you to tell me, in your mind, who will win it?
Well, it should be me.
Who deserves it?
No, we never talk about things like that.
No.
So perhaps a question from over there.
Okay, I'm Nick, I'm just a student. Do you get unwanted duplicates of strings of DNA? Like, suppose I had the string ATCG, don't know my biology, but that might pop up somewhere else completely unanticipated, and CRISPR would also target that and change that. Is that a problem?
Yes, if there's a duplicated sequence, it is a problem. I'm actually working on some genes where there's a duplicated sequence and it cuts both of them and you lose a bit in the middle. So there are a certain number of duplicated sequences in our genome and you can only cut a unique sequence. Usually you can find a unique sequence around the place because it targets about 20 base pairs. If it was just targeting 4 or 5, like you said, that would be catastrophic. And that's what we had before. Restriction enzymes could target just 4 or 6, and that was okay. But if you had a big bit of DNA, it would cut it into lots of little pieces. This cuts 20, and if you do the 4 to the power 20, that's sort of unique in the genome, except if you have a genome duplication. So it's a good question. There'll be some limitations, but it won't be too bad.
And just a quick follow-up, what happens to all the DNA after it's cut? Do you reattach something at the end?
So once anything gets cut off a chromosome, it gets degraded. So the chromosome will have to be joined up again, and the cell will stitch in any DNA it can to join that chromosome up. But any DNA that just floats around, it doesn't have a centromere, and it doesn't have origins of replication. It can't replicate. It just gets degraded. DNA floating around.
Yeah.
Thanks.
So we'll go back to this side.
Thanks. I'm Sid. I studied engineering. I don't know that much at all about molecular biology. My question relates to the questions that were asked. For CRISPR to be used in clinical settings, it has to be probably effective on, let's say, like a large number of cells, which means it needs to be delivered very effectively. And like, for example, in the skin, like get to, maybe like 70 or 90, I don't know, some percentage of cells rather than just one cell. And as far as I'm aware, like, just viral entry isn't that effective, but there are a bunch of people doing stuff related to using nanoparticles or other methods for it, or like including other DNA strands that they might want for like homologous editing afterwards. What's your view on these efforts? Is that going to be an important part of making them useful? And a second question, which kind of came up listening to other questions, was you talk about some possible fundamental limitations to how effective it could be in clinical usage. Do you think the best way to address those is to learn more about all the mechanisms of how it works? Or, for example, do like a high throughput search of like 100 different organisms that exist to explore like, different mechanisms that we haven't looked at yet? What's your take on that? Thanks. Sorry. Do we look at more things, or do we study this one better?
The more CRISPR, both,
more organisms for different systems, or just this one that we've found?
The CRISPR system probably is uncomparable. Even these other CRISPR antiviral defense systems that have been just reported are not as promising as CRISPR, just because CRISPR is unique in the fact that you can program the Cas system and the Cas proteins quite easily. So, in fact, in my lab, we are working on these issues, trying to find other variants of CRISPR-Cas that may be by nature more efficient, and they have all the properties we are looking for in these systems. I was told a few years ago that probably when you are using a CRISPR system that has been evolving for a few thousand million years in bacteria, you are not going to be able to improve that. That's not right. I think you can improve it for other purpose. So still you can improve those tools. And also we are working on a similar approach to infer how the CRISPR systems were 1,000 million years ago and 600 million years ago, and synthesize those systems. You can do that, of course, in lab and test for them. So the tools in nature are so many, and they are, in particular in CRISPR, so different one to another, that we will find the best tool there, better than these ones, and we will improve that probably through directed selection, evolution, or things like that.
So I think we're almost out of time, but what I'll try to do is just take the 3 people who are standing up. And I do think that was a very good question. I actually think getting this DNA into cells is a big issue. It's a miracle that we can get DNA into cells at all. And as a field, I just hope there's another Francisco Mojica out there working on this one, because if we could really get it into cells efficiently, that would be a big step. Yeah. So we'll go there and then these two questions. Yeah.
Yep, just a quick question. Hey, my name is Marcelo. Francisco, if you were to turn back time to just after your PhD and you had all that freedom to do no pressure research, and CRISPR was currently at the stage that it is now, what would you dedicate your time to do?
Oh, that's a good question.
So, I did a summary of that. Please help me.
What? Yeah. So would you start, if you had your career again, would you jump on the CRISPR bandwagon now, or would you say, I'm not working on that, I'm going to do something completely different?
I mean, even now, I'm sure that I will keep working on what I did. I think that we didn't expect this revolution. So it's great to enjoy this revolution. But I still believe that the most important thing in research is just to contribute to knowledge. And so basic research, from my point of view, the most important thing. And I believe in basic research. I will still keep doing exactly the same thing I did, for sure.
Thank you.
Yeah, it's a new universe. I think there's still plenty to discover.
Kumar Detal, I'm a cardiothoracic surgeon at St. Vincent's and at the Victor Chang. I've only got one question. Now, I transplant hearts and lungs, and one of the fathers of my specialty was famously known to have said xenotransplantation is a therapy for the future and will always remain so. Huh. But you, not so much with the stroke of the genius, but with 10 years of dogged scrutiny, have almost made that now achievable. What is your forecast on me being able to have readily available, safe pig hearts and lungs to transplant to my patients?
There was a huge challenge on that. That was the fact that pigs, for instance, are infected very frequently with viruses. And these are closely related to HIV virus, for instance. And not only that, but also that the number of copies of the DNA of these viruses integrated in the cells of the pigs are 60, 40, or something like that. So removing all that information, all that viral information from the cells of the pig was impossible before CRISPR. With CRISPR, researchers were able to remove 62 copies of this virus in cells. So this is a good step forward in the sense of xenotransplantation. And I know that some people in the States, it's a Spanish one, Carlos Belmonte Espizua, is trying to get organs, human organs from human cells implanted in pigs and getting organs from that. I think this is quite likely that in the future we will get organs safe organs from pigs. And not just pig organs, but human organs that have been developed in pigs. Probably that happens, I'm pretty sure.
And the last question, welcome.
Yes, hello, yes. My name is Warwick. I come from a computer background, so I don't know much about biology or medicine, but I'm very interested in this, and thank you for coming all this way. My question is, can CRISPR, because of its sort of democratization of making it so easy and cheap and all quick, be used in terrorist activities?
Yes. Yeah, you can, for instance, modify an insect to be resistant to any chemical that is being used. I'm not going to give ideas, but it's of course it's possible. But let's say, for instance, the most powerful toxin is produced by a bacteria, and you can get them just outside. Just take a little bit of earth, and you get the bacteria, you isolate it, and tomorrow we have millions of those microorganisms producing the botulinum toxin. So that we know that for many years. So it's still the possibility of using that to kill millions of people. And with CRISPR, that happens exactly the same. So it's possible to develop tools in different ways to kill people, and can be used by bioterrorists or whatever. The possibility is there, of course.
But my take on that would be that you can already grow botulism toxin, and actually, you know, the biggest threat probably at the moment is nuclear weapons. And, you know, if there were a mad dictator in the world with a choice, do I develop nuclear weapons? Do I use CRISPR? I think they'd be going for the nuclear weapon option. You know, because I think it's a sort of proven technology.
Yeah.
You know.
Sorry, just one tiny follow-up. But would the best defense against, say, a CRISPR-type attack be another CRISPR defense?
Well, that's why Francis and I are here.
Yes.
Okay, on that note,
Also,
so I'm going to enjoy some biotechnology with Francis and have a drink. Thank you all for coming. It's splendid to see you all here. I thank Francis for coming all the way down to Australia. We've worked him very hard today. It's been a wonderful day. You've been a great audience, and I just thank everyone who's been involved in the event, Meryl, the team, Vanessa, everyone.